Okay, so this is the other part of the chapter where now, instead of looking at gram-negative bacteria that we classified as proteobacteria, now we're going to move into the gram-positive bacteria, which can be classified based on the amount of G's and C's, guanine and cytosines that you have. Either you have high or low levels of these, and then if you have high levels of G's and C's, these bacteria will be classified as actinobacteria, but if they have low content of G's and C's, it will be considered firmicutes. And now something about the member of the firmicutes that it's important is mycoplasma. We have a bacteria that is called Mycoplasma pneumoniae, and this bacteria causes a type of pneumonia or infection in the lungs that is called walking pneumonia, because the person signs and symptoms of pneumonia are not as severe as the other types of pneumonia. And then one thing about this bacteria, about mycoplasma, is that it's the wall-less bacteria. So if you have a growth of this bacteria, mycoplasma, because it doesn't have a cell wall, when it grows in a petri plate, if you're looking at the top, it gives you the appearance of fried eggs. It's a pleomorphic bacteria. Remember, when a bacteria doesn't have a cell wall, it will take different shapes. And then that characteristic growth, it is definite of mycoplasmas. And then for the actinobacteria, we have several members, including Streptococcus, Staphylococcus, Enterococcus, Lactobacillus, Clostridium, Bacillus, and Streptomyces. So let's look at the individual characteristics of this. So for the bacteria in this group, so Clostridia or Clostridialis, they are bacteria that are gram-positive bacteria. They're rods, and they are anaerobes. So we have most members of Clostridia that are pathogens. So we have C. tetani, causes tetanus, which is spastic paralysis. In this case, the muscles are very hard, they don't relax, And then the person's, when it has the infection going into the respiratory muscles or even into the pharynx, the person will die of asphyxia. And then when the infection reaches the muscles, or when it affects the muscles of mastication, the persons cannot open the jaw. We have Clostridium botulinum, which causes the opposite. So these bacteria causes flaccid paralysis or botulism. So remember what I told you before about the floppy baby syndrome. When the baby ingests endospores of this bacteria, the bacteria grows in the body of the baby, releases a toxin, botulinum toxin, which you know as Botox. And then the muscles of the baby, the ones here in the neck area, they cannot contract, they're flaccid. And then if the baby already was able to maintain its head erect, the baby will just go like this. That's why it's called floppy baby syndrome. Clostridium perfringens causes gas gangrene. So this will be a disease of the subcutaneous tissue, an infection. So the bacteria with the spores of the bacteria enters into a wound, and then these spores germinate under the skin, in the subcutaneous tissue, in the hypodermis, and then they start destroying the tissue. And then since these bacteria are anaerobe and they release gas, they create bullae, like blisters, but big blisters in the skin. And then when one of these ruptures, like if you have an open wound and it ruptures, the smell from that gas that exits is very bad. And then we have C. difficile, or C. diff, that causes pseudomembranous colitis. Or pseudomembranous. Colitis is an infection of the colon or the large intestine. So this infection happens when someone receives antibiotics in the hospital, like 7 to 14 days, especially the elderly. All the normal flora from the large intestine goes away because antibiotics don't think they only kill pathogens. They kill also microorganisms from the normal flora. So in this case, when the normal flora is gone, since C. difficile is part of the normal flora of the large intestine, it starts growing much faster than the rest of the bacteria. And as it's growing, it produces like fibrin type of tissue in it, and that's why it forms like a pseudomembrane. So the lining of the stomach, instead of, of the intestines, instead of having like this normal epithelium, which is simple columnar with cilia, then, or with mucous cells, then you will have like a flat surface. And then when the person is ingesting something, well, that food is not absorbed, and also water is not absorbed, so the person will have severe diarrhea. There are certain antibiotics that are more prone to produce this type of problem, like clindamycin. And then doctors, they already know, so they try to avoid giving this type of antibiotics. So that's for Clostridia. Now for Bacillales, we have many members like Bacillus cereus, Bacillus anthracis that are pathogens, and they are rods. Remember, Bacillus means rod, and they are gram-positive. So we're looking now at gram-positive, which stains purple with the gram stain technique. And then Lactobacillus will be bacillus or rods that produces lactic acid. And we have lactic acid bacteria normally in our body, like in our oral cavity, in our intestines, in the vagina. Why? Because we try to acidify the pH to prevent infections with pathogens. And then lactobacillus in the industry can be used to produce yogurt. And this can be actually part of these probiotics that can be ingested in order to actually help for the person to have normal flora in the intestines and have better digestion. Which actually this is what also can be done with patients who have pseudomembranous colitis. They can give them probiotics so that they can regain the normal flora much easier and they don't have any problems. And then Mycoplasma thalis will be Mycoplasma, the one that we saw before, and then we have the actinobacteria with different members. So here we have a dichotomous tree of the gram-positive bacteria that are lows for G's and C's. So remember, in science, dichotomous trees helps to classify organisms. An example, let's say, even for us, for humans, let's say that we're trying to classify humans based on diet. Well, you can have vegetarians, non-vegetarians. That's a dichotomous tree. And then from non-vegetarians, well, you can classify them as those ones that only eat red meat or those ones that eat both red meat and white meat, for instance. The non-vegetarians, well, can be fruity, well, the ones that only eat fruits, or well, they can be also subclassified as vegans or vegetarians, and then for vegetarians, some that eat eggs, ovo-vegetarians, or the ones that eat fish and vegetables, which will be pesci vegetarian. So we can start doing that with this dichotomous tree. So this is what you're looking here again. So here we have then low G's and C's gram-positives. You can classify them as wall-less, like Mycoplasma thalis, or they may have a cell wall. And then for those ones who have a cell wall, can produce endospores or do not produce endospores. The ones that do not produce endospores are Lactobacillus, and the ones that produce endospores can be anaerobic, like in the case of Clostridia, or aerobic or facultative anaerobes, like the Bacillus. So that's the dichotomous tree for low G's and C's. So here are the details of this Clostridia. So Clostridia, again, they're anaerobes, and they are obligate anaerobes. Remember, obligate anaerobes will be those microorganisms that will be killed by the presence of oxygen. So where you expect them then to be, or to thrive, will be in areas where you have low contents of oxygen. And then Clostridium perfringis, besides producing gas gangrene, it produces foodborne diarrhea. And then again, they are gram-positive rods. Now, Epulopicium is a bacteria from the environment. It is a symbiont with Red Sea surgeonfish. Remember, symbionts means that you have a microorganism, or well, not a microorganism, you have two organisms living together in which both benefits. So a polypicium benefits from living with this Red Sea surgeonfish, and Red Sea surgeonfish benefits from having a polypicium. Now, here we have a scanning electron micrograph of C. difficile, in which you have this rod, and this rod is producing endospores. These endospores are important in diagnosis. Remember, endospores are structures that are formed when the bacteria is about to die, and then it releases them into the environment as a form of survival. So this vegetative form then sporulates to form these endospores. And I told you about this, that it's important for diagnosis, because when someone has an anaerobic infection, and then they make this gram staining technique, and it turns out to be a positive rod with terminal endospores, that it is C. difficile. Some Clostridia might have the endospores here in the center, so that will be central. And some of them, they have it just below the end, so that will be subterminal. And that's how you can distinguish whether it is C. difficile, Clostridium tetani, or botulinum. And then here we have this cigar-shaped polypicium. It is actually a giant prokaryote. So this is a protozoan, paramecium. It's a free-living protozoan. It lives in lakes, this, or in ponds of water, this paramecium. And this is a large cell. So look here at this scale bar. So more or less, this is around 100 micrometers. So this paramecium measures more or less like almost 200 micrometers. So this bacteria, it will measure like 4 to 5 times this. So it's very large. Typically, the largest size will be like 100 microns for the bacteria. But this is a gigantic prokaryote. So what it tells you, just like as mycoplasma doesn't have a cell wall, and we typically encounter that in microbiology, there are always exceptions to the rule. So someone may think, well, this can be an algae or some other microorganism, but not bacteria. Well, in the case of this is a bacteria, no matter, it's a giant, because according to its ribosomal RNA, it is a bacteria. And now, when it comes to these bacillus, bacillus are endospore-producing rods, and they are not motile. So they don't have flagella to move, and they are facultative anaerobes. Facultative anaerobes means that, again, they can live with or without oxygen, but they prefer to live in the presence of oxygen. And then we have Bacillus cereus that produces food poisoning. Typically, it is contaminating rice. And we have Bacillus anthracis that causes anthrax. Anthrax is a zoonotic disease. It can infect cattle It can infect humans. And these Bacillus anthracis can be used as a bioterrorist weapon. So it causes severe infections in the skin that causes like ulcers, and it's highly contagious. It can kill persons right away. Staphylococcus, it's a grape-like cluster of cocci. You saw those drawings that I made yesterday in the lab. And they are facultative anaerobes, and they're halophiles. What are halophiles? If you know what it's a halophile, you will use it for a way to grow this bacteria in the lab. So what is a halophile? Philos means like, right? Halo. Philos is loving, like. Loves what? Halloween? Salt. Yes, so a halophile loves salt. So in this case, Staphylococcus is a bacteria that lives in an area that can live in areas that is having high contents of salt. In one of the ways to preserve food, I told you this before, is to adding a lot of salt. But nowadays, of course, we use that not only to preserve food, but as type of a flavorant. And then beef jerky, some fish, even shrimp can be salted and then be served like that. Like smoked fish, for instance, is having a lot of salt content. And then this is one of the bacteria that potentially can contaminate these foods. As long as these foods are not contaminated with bacteria, can be preserved with salt, and that's fine. But again, if whoever is preparing it is not careful enough, he has Staph aureus in his hands, then he's going to contaminate the food, and then Staph aureus will be growing a lot like crazy. Now, this bacteria, when it grows in agar, produces a yellow pigment. That's why it's called Staph aureus. Aureus, it is one of these words from Latin that means gold. So here we have in this periodic table, Au, that's the abbreviation for gold. And then when it contaminates food, it can produce food poisoning, toxic shock syndrome, and infection of internal organs like the heart, like our bone marrow. Okay, so this bacteria produces serious infections and intoxication. So here we have the growing of a bacteria out of this endospore. So this endospore was released into the environment, and now this bacteria grows. This is a rod. So it is turning into a vegetative form. Now here we have Staph aureus. This is an scanning electron micrograph, same as the previous, well, this one is a transmission electron micrograph, and this is a scanning electron micrograph. So in this case, you can see the cluster of bacteria, and this color is enhanced with a computer. Okay. But something to keep in mind about this Staphylococcus is that it has a cluster arrangement. Staphylo means grapes in Greek. And then for lactobacillis, they are air-tolerant anaerobes. So they are mainly anaerobes, but they can live in small or minute amounts of oxygen. That's why they are air-tolerant. And they don't have an electron transport chain. That's why they are air tolerant. So, and they are lactin producing bacteria, as I told you before. Members of these lactobacillus will be lactobacillus itself. So this is how it looks. A streptococcus, enterococcus, and listeria. Listeria, it can cause stillbirth. It is found to contaminate foods like vegetables, dairy, delicatessen, preserved foods like hot dogs, etc. And these bacteria, Listeria monocytogenes, can survive fridge temperatures. So then many of us will buy food and think about that it is safe to consume that food. We put it in the fridge, but then after that we can have listeriosis. And now this bacteria also, it is found to have been contaminating salads that are bought in store. And then whenever they find these contaminants, they make huge recalls of these foods. And it's a public health problem, actually, this type of bacteria. Enterococcus produces infections in hospitals, so it's a nosocomial infection. And the streptococcus, we have different types that we classify according to the antigens. We have the Lancefield classification, which is like the most famous one, and then we can classify them with different letters, and some of them are important human pathogens, like these ones that we have here. So here is the location for lactobacillus. I already told you, vagina, intestinal tract, and oral cavity. And this is part of the normal flora for protection for lactobacillus. And then for streptococcus, it's chains of cocci. So we have Streptococcus pyogenes, the one that typically produces pharyngitis. And this one is beta hemolytic. So what it means is that when you grow it in a plate with blood agar, and these erythrocytes from this agar come from animals like sheep blood. So all you do is just remove the fibrin, and then you add it into nutrient agar or any other solidifying agent, and you make, with sheep blood, you make these agar plates. So when this bacteria grows in the agar, it produces a white halo around this agar. So if this was red color, you have solid red color because you have your agar plate, and then you have growth of Strep pyogenes here, a colony. This white halo means that you have complete hemolysis or hemolysis. This is known as beta. So this is a clear halo. Which, I mean, a halo is that, right? It's clear area. Clear area / halo around S. pyogenes. So these bacteria, besides causing pharyngitis, causes scarlet fever, impetigo, and rheumatic fever in the long run. This impetigo, it's an infection in the skin, and then for rheumatic fever, it's a sequelae. A sequelae, it's a consequence that happens after having repetitive infections. So in this case, you will have partially some injury to the articulations. And also, you can have problems in your heart valves, and you need to have replacement of these heart valves. And this happens, these infections with strep pyogenes pharyngitis happens frequently during childhood years. And then with time, the person with years will develop these sequelae, like in adulthood. And then Streptococcus agalactiae causes neonatal sepsis. So the baby born, and then it will have serious infection after it borns. And then we have strep pneumoniae that causes strep pneumonia. Its name tells you. And then Streptococcus mutans causes caries. And Enterococcus causes nosocomial infections. Listeria monocytogenes causes stillbirth. So see, gram-positive bacteria can cause from mild injury, which is not so mild. I won't say caries is mild, but it can cause caries, and then it can cause death. Mycoplasma, it is very small, measured between 0.1 to 0.25 micrometers. So then, this mycoplasma can contaminate sources of water, for instance, if it's not filtrated properly with a filter that traps something smaller than 0.25 microns or 0.1 microns, even it can pass through filters. And this is what I told you before, causes a typical pneumonia or walking pneumonia and gives you fried egg shaped colonies. So this is how mycoplasma looks like. This is an scanning electron micrograph. It has these terminal structures. So this helps for the bacteria to adhere to surfaces, and then when it grows, it creates filaments. So this organism actually reproduces by fragmentation of this filament at the bulges. So this is areas where it starts fragmenting. So those were the low G's and C's gram-positive bacteria. Now let's look at the high G's and C's. So high G's and C's can be subclassified as bacteria that forms filaments or the ones that they don't form filaments. Another name for filaments is called mycelia. So this one have mycelia, these ones, they don't have mycelia. Now for the ones that have mycelia, either cannot have conidiospores or they can have conidiospores. And conidiospores is just a form of growth of these spores. It's just how you call them, just like the conidiospores that you saw in these Myxococcalis. And then for those ones that they don't form conidiospores, they can be acid-fast, like Nocardia, or they can fix nitrogen, like Frankia. Now, for the ones that forms conidiospores, they can be aerobes or anaerobes. If it's an anaerobe, will be streptomyces. If it's an anaerobe, will be actinomyces. And then for the ones that they don't form mycelia, they can either be acid-fast, like Mycobacterium, or not acid-fast. If you remember, acid-fast is a staining technique in which you have a primary dye, which will be carbolfuchsin, and then you have a decolorizing agent, which is acid alcohol. So it's alcohol with hydrochloric acid. And then bacteria that resist the decolorization with acid-fast will be acid-fast. And the ones that decolorizes will be not acid-fast. And then for these ones that are not acid-fast, you have to add a secondary stain, which will be methylene blue, to see the bacteria. Otherwise, you won't see it. It's like safranin for gram stain. And then for the not acid-fast bacteria, either can stain gram positive or gram negatives. If it's gram negative, will be Gardnerella. And then if it's gram positive, can cause diphtheria like Corynebacterium, or can make propionic acid like Propionibacterium. So here are then the members of actinobacteria, from actinomyces up to streptomyces. Streptomyces has filaments. And then within these filaments, it has coils. So it branches into coils, and these coils have little sphere-like structures that we call the conidiospores. So this is a scanning electron micrograph of these streptomyces with these filaments and these coils. And in these coils, that's where they have these conidiospores. And then here we have actinomyces, that it has filaments that branches, but you don't see the coils, and you don't have conidiospores for this bacteria. So now we're going to see characteristics of gram-negative bacteria that are not classified as proteobacteria. So we're going to see Bacteroidetes, Cytophaga, Fusobacteria, and some bacteria that are environmental, like green sulfur, green non-sulfur, purple non-sulfur. And then we're going to see as well some bacteria like spirochetes and Deinococcus. So within the microbial world, we have some bacteria that live in the environment that are phototrophic. They use the sunlight to generate energy. And then out of these bacteria that are phototrophic, some can produce oxygen and some cannot produce oxygen. So that's why you call them oxygenic photosynthetic or anoxygenic photosynthetic. So here's the chemical reactions for this. So here we have water combined with CO2 plus light. That gives you carbohydrates, water, and oxygen. So this is oxygenic photosynthesis. And this is what happens with many microorganisms and even with plants. And algae, for instance, is one of these microorganisms that is oxygenic photosynthetic. So thankfully, we have these organisms like plants and these bacteria because they can absorb the CO2 that we produce with our cars and industry. So they clean the air. So they remove the toxic gas, which is CO2, and they give us oxygen. Photosynthesis is very good. So plants are good for us. And then some bacteria or some microorganisms can combine hydrogen sulfide, which is also a toxic gas for our environment, with CO2, and with the aid of light, They produce carbohydrates, water, and elemental sulfur. This one can be used by plants. And this is anoxygenic because you are not producing oxygen, you are producing elemental sulfur. Okay, so these are the two different types of photosynthesis. So then we have oxygenic photosynthetic bacteria and anoxygenic. So here is a table that combines both oxygenic and anoxygenic photosynthetic bacteria. So here's the common name, here's the phylum, examples of members, and then here we have what is the electron donor for reduction of CO2. Anytime that you have these different types of bacteria, you can expect that they will use photosynthesis, but some of them, they can produce oxygen and some of them cannot. Which ones can produce oxygen? Well, it will be cyanobacteria, like Anabaina, and the electron donor for CO2 reduction will be water, so then it produces oxygen. And then for green sulfur bacteria and purple sulfur bacteria, you have this electron donor for CO2 reduction, which is hydrogen sulfide. So this bacteria will be anoxygenic. Purple sulfur and green sulfur bacteria. Example of green sulfur will be chlorobi, and then for purple sulfur will be chromatium. Now, we have green non-sulfur and purple non-sulfur bacteria that they have an electron acceptor, which or donor, which will be organic compounds for both, and both are anoxygenic. Example of green sulfur bacteria, chloroflexi, and for green sulfur, purple non-sulfur will be rhodosporillum, which is a proteobacteria. And then bacteria like cyanobacteria fixes nitrogen, and it is gliding. Many of these bacteria live in aquatic environments like cyanobacterium, and they form these filaments. It's a rod, and they form heterocysts. So these heterocysts, like you can see them in this case for this bacteria, which is Gloriocapsa, this bacteria has a capsule and it holds these heterocysts internally. So this bacteria divides by binary fission, and it is held together by this capsule, which is made out of a glycocalyx. And it's a unicellular non-filamentous cyanobacterium versus this one, which is filamentous. So you can have two types of cyanobacteria, the one that form filaments and the ones that they don't form filaments. Now, for chlamydia are bacteria that lives inside cells, so it's an intracellular parasite. We have different species that are important. For instance, we have Chlamydia trachomatis that causes trachoma, also causes sexually transmitted infections and urethritis. This one is the #1 cause of STDs in the United States, and it is this bacteria that potentially can lead to ectopic pregnancy. How does that happen? Well, when this bacteria grows in the reproductive tract of females, it will burst the cells from the lower reproductive tract, So let's say this is the vagina, and then in here you have the uterus. And this is the entrance for the uterus. So this is the cervical canal here. So when this bacteria goes up like this, and then here you have the fallopian tubes, this bacteria crawls up and then when it enters here into one of these fallopian tubes, it starts destroying this epithelium and it will cause a scar. And that is going to lead to an obstruction. So if you make a section in here and you see it sideways, the lumen of the fallopian tube is going to be occluded. So this is a scar tissue. And then the egg cell, which is a gigantic cell in comparison to the sperm cell, will be ovulated and it will get stuck in here. You cannot go into the uterus. But the sperm cell, which is small, maybe it can go through here into a tiny opening in this scar tissue, so it can go through this. And then it fertilizes the egg, and this is not good because once the zygote starts growing, there is no space here for the zygote to grow. And then you will have a rupture. So we have a space here in the uterus for implantation and for growth of the fetus, but not this place. So then, if this ruptures, it's a medical emergency, there will be a hemorrhage, and then the mother can potentially die. So that happens in the female reproductive system. Now, in the male reproductive system, something similar happens. So the males have the vas deferens, which is the tube that brings the sperm into the outside of the body. But when this bacteria infects this vas deferens, so this bacteria will produce scars here, and then the sperm will not pass there. So the lumen is going to be similar as to this. If you make a section like this, you see the lumen of the vas deferens will be like this. And some sperms can pass, but most of them not if it's partially obstructed, but more than likely this is going to be completely obstructed because it's very tiny. So then the male will be sterile. So actually, this is one of the causes of infertility too. And that will be for both males and females. Like, let's say the bacteria, it is infecting the female reproductive tract. This is the vagina, this is the uterus. The bacteria don't think that it is going to go to only one side. It will go to both. So if this is the right fallopian tube and this is the left, it can do the same in this area. So then it can lead to infertility because let's say that the female had only one side obstructed for ectopic pregnancy. What if this other one is not obstructed? Well, she can still have fertilization because she will have one egg going into this area. But sometimes when this happens, doctors ask, and then both fallopian tubes can be tightened and gone away. It would be easier to say, well, I want to get pregnant again, and then just remove the obstruction and try to tighten the fallopian tube, but it's not as easy as it sounds. So then it can lead to reproductive problems, this type of infection with Chlamydia trachomatis. Now, Chlamydophila pneumoniae, which is another type of chlamydia, causes atypical pneumonia. And then we have Chlamydophila psittaci that causes psittacosis, which is an infection in the lungs, and that it is produced when someone who handles birds or someone who get in contact with droppings of birds, and these droppings of birds have chlamydia or Chlamydophila psittaci, and the person will develop pneumonia. Okay? Now, many birds' droppings have not only chlamydia pneumonia, Chlamydophila psittaci, but also can have other microorganisms like Histoplasma capsulatum. And we don't live in this area of the nation where we have this type of infections. There are certain areas, certain states where you have these type of infections more prevalent. However, something at least that I do is when I'm walking in the sidewalk or in the street, if I see a bird dropping, don't think that I will step on it. I will, even if it's dry, I will just do this. So if you see me doing this when I'm walking, no, I'm not drunk. I'm just avoiding stepping on this. That's one thing. Now another thing is, in those areas where you have birds with these droppings that has Chlamydophila psittaci or Histoplasma capsulatum, birds drop their droppings in the street, right? And they dry out eventually with time. But there's a wind in one of these windy days, and people is talking or eating something, and as they talking and eating something, the wind comes into their face and then to the mouth, and then the microorganisms goes into the body. It's a good idea of not talking so much when it's a windy day. That's all that I can recommend. And this is the life cycle of chlamydia. So chlamydia has an effective structure that is called elementary body. The elementary body binds into the plasma membrane of the host cell, and this will be endocytosed, so you will have vacuole with this elementary body, and then this elementary body will reorganize to form a reticulate body, which is larger than the elementary body, as you can see. And then this reticulate body starts dividing and forming multiple reticulate bodies. It takes around 48 hours for this to happen. So then these reticulate bodies start dehydrating, forming elementary bodies, and then after you have many elementary bodies ready in this vacuole, then you will burst the cells, and then you will see more cells that are uninfected. So that's what happens in this area. So let's say that the infection starts here in the epithelium of the vagina. So these are the cells of the vagina, and then they get infected. Within 48 hours, you will have multiple reticulate bodies in this epithelium, and these cells will burst and release all of these reticulate bodies into the lumen of the vagina, and these ones will go up, then contaminate the lining of the uterus. You will have more reticulate bodies, and then that's how they sit up, going up. And that happens as well in the male reproductive tract. So it's a life cycle. So this is a micrograph of Chlamydia psittaci. So this is the elementary body, reticulate body, and something between reticulate and elementary body will be the intermediate bodies. It has a dark center, the intermediate bodies. And now we have some bacteria that it's one of the exceptions to the rule. Remember, bacteria are prokaryotes. Prokaryotes, they don't have a nucleus, which means that they don't have a nuclear membrane that surrounds the DNA. But what you can see here, well, this is a nuclear envelope in this bacteria. This is Gemata obscuri globus. So it has a double membrane around the DNA, which is the nucleoid. So this is one of the exceptions to the rule. This is a Plantomycetes. Another non-proteobacteria, it's a gram-negative, will be Bacteroidetes, which are anaerobes. And these Bacteroidetes are found in the mouth and in the large intestines. And Cytophaga degrades cellulose in soil. So it's part of this, it's one of these members of Bacteroidetes, degrades cellulose in soil. And then Fusobacterium, it is another gram-negative, it's an anaerobe, and are found in the mouth, and together with Streptococcus mutans, they form like little groups. They can be involved in forming caries. So it's one of these bacteria that can cause us also dental problems. And this is Fusobacterium, like this one. It looks like a spindle. So we already saw this classification of the bacteria that are anoxygenic photosynthetic. The oxygenic photosynthetic, remember, is cyanobacteria. So we have purple sulfur, green sulfur that uses hydrogen sulfide as an electron acceptor, while purple non-sulfur and green non-sulfur uses the organic compounds as electron acceptors. And they don't use, they don't produce oxygen. So this is how purple sulfur bacteria looks like. So it has this nice coloring because it has different levels of oxidation of sulfur. Now, a bacteria that or a couple of bacteria that has been important in research, and we're studying them for a reason, are Deinococcus, radiodurans, and Thermophilus aquaticus. Deinococcus radiodurans have some enzymes that are capable of swapping pieces of DNA that is mutated, like recombinase enzymes. And this bacteria is resistant to radiation more than endospores. That's why it's called radiodurans. Durans for duration, radios for radiation. These bacteria, with these different recombinases, again, can swap pieces. You subject these bacteria to radiation, and then you have a mutation in the DNA, and that mutation can repair it. So we're trying to see if with these enzymes, these recombinases, we can do the same for humans. If we have a mutation in our DNA, if we can swap it for a normal piece of DNA. And now Thermophilus aquaticus, it's a bacteria that it is a hyperthermophile. It was found originally in hot springs in Yellowstone National Park. And this bacteria has an enzyme that is called Taq polymerase, and it is Taq polymerase for Thermophilus aquaticus. This enzyme can amplify pieces of DNA really quick because you can separate the two strands of DNA with high temperature, and this enzyme can start forming a new strand of DNA from these two strands that you separated. And this is one of the enzymes that it is used in polymerase chain reaction technique, or PCR. And it is one of these enzymes that it is used to amplify a tiny piece of DNA to make multiple copies to make diagnosis. Now for the Archaea, we have extreme hyperthermophiles like Pyrodictium, Sulfolobus, and then we have methanogens. So this is the classification of these Archaea, which are extremophiles. And then we have methanogens that lives where methane gas is produced, like Methanobacterium. And then we have Halobacterium that lives in areas where you have high salt concentration, like for instance in the Dead Sea. So what are the highlights for this chapter? Well, we have mutations in horizontal gene transfer, and that makes these bacteria to be of different kinds. And bacteria, as we saw, they live in different environments, from areas where you have a lot of salt, areas where you have low nutrients, areas where you have sulfur, etc. And this has selected for a huge diversity of microorganisms. So microorganisms, in general, you can say that they adapt to different environments.